particular discussing the challenges and the issues around developing quality products from the plant to the finished product, the tablet that goes to the patient. I will use two case studies. The two case studies are two products, Ginkgo-san and Remotiv. They are both products promoted by Flordis, and they are both for brain functions. But let's start from the real beginning with a very interesting example. This is really our trademark, is our IP on this example. I'm just joking because Professor Stau claims that it's his own baby, this example, and it's possibly true. He has an amazing knowledge about wine, so I guess it's possibly true. But in a country where wine is so important, like here, and you have some of the best wines in the world, I think the concept is very clear. You could have same Cabernet name, but in the end you can have two different bottles, a very expensive and delicious wine, or a cheap cask wine that is only good as a table wine or to cook. This depends on the way the plant, the vine plant has been grown, the way the soil condition, the cultivation condition, the manipulation of the vine, the time the grapes have been collected. If you collect the grapes during a hot, sunny day, you have higher content of sugar and therefore a more alcoholic wine, etc. Medicinal plants are not so different. They actually offer even more challenges. So let's see a little bit more in detail the challenges that we face from the plant to the finished product. The first one is obviously the plant itself, the genus, the species of the plant. Each species of plant have different subspecies. Depending on the subspecies, you might have different content of active components. How the plant has been growth, the soil condition, the chemicals that have been added to improve the growing, the method of cultivation, and then harvesting and storage. What part of the year the plant has been harvested, at which stage of the growth cycle, how the raw material has been stored and packaged. And then, of course, the extraction, how the active ingredients have been extracted, how they've been, which kind of equipment have been used, how they've been standardized for which products, which concentration protocols have been followed. And finally, the finished product, either a tablet or a liquid or a powder. It depends on the formulation, manufacturing procedures, the excipients, the dissolution time, how it's been stored. All these stages represent a massive challenge, but all these stages are controllable. It is possible to reach a standardized journey from the plant to the finished product. If you manage to control all this variability, you have a medicinal formulation that then can be used in clinical trials. This has two main advantages. The first one is the reproducibility. So you have a product that is always the same, and the patient is assured that when you support your product, the claim of your product with a publication, the safety and efficacy is exactly the same as the one proposed in the clinical published trials. Just a couple of examples. In this case, anthraquinone is active component of rhubarb, is a laxative plant, planted for use as a laxative. The active components, if you harvest the root after 2 years, are much more than if you harvest only after 1 year. So a completely different therapeutic profile. Valerian. Valerian is a very common plant for sleep induction as a relaxant. This plant has a 51 weeks growth cycle. If you collect the plant at different weeks, you have a completely different content of the active ingredients of interest for therapeutic purposes. So you might have a product that works or a product that does not work.